Now that we understand the concept of Gibbs free energy in the context of chemical reactions, let us investigate a special type of chemical reaction that happens in cells that involves the energy currency of the cell, or ATP. So we can see here in this reaction that ATP, in order for it to provide energy to our cells, needs to be separated into two molecules, ADP and an inorganic phosphate. And water is needed in order for this reaction to happen. Notice that the free energy of this reaction is negative 7.3 kilocalories per mole, meaning to say that this reaction in the cell is very much spontaneous, or is very much favorable in terms of thermodynamics. We also take note the equilibrium constant of this reaction. Take note here, the concentration of ADP must be really high as compared to the concentration of ATP in order for the equilibrium constant to be reached in this particular reaction. Meaning to say that if there are already a lot of ADP and very few ATP, this reaction is going to reach equilibrium. Meaning to say there will be no visible reaction happening. But we don't want that in our cells. In fact, our cells strive so that the reaction that we see here, ATP becoming ADP, is favorable. So how do we keep the environment in the cell so that this reaction remains favorable? In other words, how do we prevent it reaching its equilibrium constant? Well, simple. If the equilibrium constant is when ADP is a lot higher in concentration than ATP, then the cell maintains a high ATP to ADP ratio. Meaning to say our cells are working overtime to produce more ATP than the ADP that it can produce as a result of the hydrolysis of ATP. Now that makes sense. And where does the cell get the energy to make more ATP? We already know it's from the food that we eat. So here's the structure of ATP as shown here on the left. So it's a very energy-rich molecule, particularly on the last bond between the inorganic phosphate, the third inorganic phosphate, and the second inorganic phosphate. Okay, so we have the triphosphates here, which are all negatively charged, which results in them storing a type of energy we call chemical potential energy, which is broken or released when the last phosphate group is cleaved away from the other two phosphates with the help of water. Okay, so we can see here that this reaction is reversible. However, the favorable direction is going to the right, wherein ATP becomes ADP. And again, the cell achieves this because the cell doubles its time in making more ATP than the ADP that's accumulating in it. So here's one of the other chemical reactions that the cell needs to do. Okay? In this case, the creation or the making of glutamine. Okay, this is an amino acid from the raw materials glutamic acid and ammonia. Now, in order for the cell to do this, there is an enzyme that's used to catalyze the reaction. It's called glutamine synthetase. However, notice here that the free energy of this chemical reaction is positive 3.4 kilocalories per mole. Meaning to say, this reaction is not spontaneous. It's like cooking an egg. Meaning to say, you cannot just do nothing and wait for the egg to cook. You need a source of energy for this to happen. So, the question is, how would the cell make this possible if this reaction is non-spontaneous? The answer is, it needs a source of energy to be coupled with this particular reaction. Okay? And that source of energy that's going to be coupled with this reaction is that of ATP. So we couple it with the hydrolysis of ATP, which has a free energy change of negative 7.3 kilocalories per mole. So this is how the cell does chemical reactions that are unfavorable, okay, in producing important chemical compounds like glutamine. So the cell always couples it with a favorable reaction, in this case, the hydrolysis of ATP. So how does that happen? So here in the reaction here on top, we can see that ATP actually donates its last phosphate group to glutamic acid. So this process, we are going to be encountering this again very much all of the time in the next topics in our cell and mole lecture. This process, when ATP gives its phosphate group to another molecule, is called phosphorylation. Okay? So ATP gives its phosphate group to glutamic acid, in the process becoming ADP. And this is a free energy change of negative 7.3. That's favorable. Now that glutamic acid is energized because it got the phosphate group, it can now react with the ammonia, and together they form glutamine. And in the process, it releases back the P, or the phosphate, that was given to it by ATP. And this reaction of ammonia plus glutamic acid is unfavorable with a Gibbs free energy of change of positive 3.4. And this happens with the help of an enzyme, glutamine synthetase. So now, if we're going to total the free energy changes of these two reactions now that are coupled, the hydrolysis of ATP and the synthesis of glutamine, the overall change in free energy is now negative 3.9. And this means that this reaction is now favorable, and the cell is able to make glutamine from glutamic acid and ammonia with the help of coupling that reaction to the hydrolysis of ATP. So this is the key, guys, the coupling of reactions. So we need to define two terms here, equilibrium and steady state. A cell or a system that is in equilibrium, okay, is said to be dead. Meaning to say, there is already a random distribution of materials and molecules inside the cell because the cell has lost the life force to actually organize its internal structure. Notice here that the amount of ADP is so high and the amount of ATP is so little. The reaction of ATP becoming ADP has been reached here in equilibrium. There is no more reaction and the cell is dead. You would never want your cells to be in equilibrium, guys. However, there is another condition of the cell we call steady state. And in steady state, the cell is constantly working to keep its internal environment stable, steady. Okay? So don't ever mistake the stable internal environment of a cell as equilibrium. The cell actually attained that stability by working double time. of course, with the help of ATP. As you can see here on the graph on the right, in a steady state cell, the amount of ATP is constantly being replenished so that it's so much higher than ADP. So there is no equilibrium here, and the cell is able to do a lot of work because there's more ATP than ADP. So this is what we want our cells to be in steady state. So here are some of the roles of ATP in the cell. Okay? So a while ago, we have seen how ATP is coupled with certain reactions that are not spontaneous so that the cell can make them happen. The cell may also use ATP, for example, in letter A, in maintaining the membrane potential, especially in neurons or nerve cells. In letter B, we can see here the cell using ATP in order to move potassium ions into the cell. In letter D here, we can see ATP being used by muscle cells to contract for movement. And in letter E, we can see ATP here being used in the phosphorylation of certain proteins that is usually happening during cell signaling.